The title of this, why don't we call it How to Do Genetic Crosses. Okay. The first example that I'll use is a true breeding or purebred, true breeding tall plant, and this is a tall pea plant, is crossed with a true breeding or purebred small plant. And if you look at these pea plants, they have a lot of different characteristics, as we've seen. Some have flowers that are axial on the stem. Some have flowers on the end. But in this case, we have a true breeding, and we're looking at height. Tall plant. And here's the short plant. So we're looking at the phenotype, and one is short and one is tall. Now, because it's true breeding or purebred, we can tell the genotype. The phenotype is what it looks like. The genotype is the alleles. So we usually use the first letter of the dominant trait. So tall is dominant, so we're going to use T. So this one's true breeding tall. So we're going to use big T for that. And true breeding small is little t, little t. Now, the reason I knew that was because in the problem, I would have had to say that all the offspring are tall. And plus I gave you Mendel's genetic traits, and I let you know on that sheet which ones were the dominant traits, and tall was the dominant one. I probably won't ask you to memorize that, but you should have a good idea of which ones are dominant, which ones are recessive. But I will always give you a clue in the word problem as to which one's dominant and which one's recessive. So because I know that all the offspring are tall, I can automatically know that if I breed a true breeding tall with a true breeding short and all the offspring are tall, I know that tall is the dominant trait. So that's why I chose big T for tall. Well, let's do the cross now and see what that looks like. This is a Punnett square, and remember, Punnett is that English word, Old English word for a sectioned off carrier for fruit that they used to use back in the old days when they used to deliver fruit to people's houses and the store and whatever. And a punnet was the carrier, the wooden box where they put the apples or the peaches or whatever. So we're going to have big T on one side. It doesn't matter which side we use for this because we don't know which one is male and which one is female. Usually, if we're doing human genetics, the male goes on the upper side and the female goes on the right hand or the left hand side, just like we saw in that video. So little t goes over here, and combining the top and the side for each one of these squares, we get big T little t there, big T little t here, big T little t here, and big T little t here. Well, automatically we know that all of these are 100% tall, and they're referred to as heterozygous. 100%, sorry. Heterozygous. Remember that heterozygous means that they have one dominant allele and one recessive allele. This one over here would be called homozygous dominant. And this one here would be homozygous recessive. I want you to know that, so write that down. Write this down. Write this down. Homozygous recessive, homozygous dominant, and heterozygous. I will expect you to know that for the test. Okay? Are we good up to this point? Okay, a little longer. I'll wait a minute before I turn the page. Actually, I can do this. Let me just go up here and do this so I can carry on here. So, let's now take these plants, these heterozygous plants, the offspring, in our example from 5.1 in the review and reinforce. They then said take 2 of the offspring and cross them together. Well, 2 of the offspring means 2 heterozygous plants and cross them together. In fact, this plant would produce seeds that have a big T allele. Here's a P with a big T allele. Here's a P with a little t allele. All these P's have little t or big T. Likewise, this plant produces P's, 1 big allele and 1 small allele in its P's. These are actual peas that you would eat from the pod. This might be a single pod right here. Here's the pod, the pea pod. And some of those peas have the allele on the chromosome for little t. Some of those peas have the allele on their chromosomes for big T. Well, we don't know which of these peas are going to cross when the plant is planted and fertilized, but here we go, and we'll see. So we have a big T here, a little t here, a big T here, and a little t here. So what do we get? Well, we have a big T, that's homozygous dominant. We have a big T, little t here, a big T, little t here, and a little t here. If we analyze the genotypes, homozygous dominant, there's going to be 25% of the offspring are that. Big T, little t, the heterozygous ones, 50% chance of having that. And little t, we have a 25% chance of the offspring being that. That's the genotype. The phenotype looks a little different. In the phenotype, there's going to be 75% of the offspring are going to be tall. 25% of the offspring are going to be short. Okay? Does everybody see how that works? Okay. And this is what Gregor Mendel did. He crossed pea plants, 6,000 different pea plant crossings he did. And he found that when he crossed true breeding tall with true breeding short, he always got tall as the offspring. And then when he crossed those offspring, the ratio was always 3 to 1. 3 tall to 1 short, or a 75% to 25% ratio. Okay? Does that make sense? There could have been 30 tall plants and 10 short plants. You just scale up the ratio depending on the number of plants you plant. Okay? Well, that's with pea plants. What about humans? What about human traits? And even one that we've already taken a look at. Let's see what that looks like. We'll do one cross with humans and see how that looks. And we'll use the characteristic of earlobe shape. So a homozygous dominant man has free earlobes. Homozygous dominant man has free earlobes. A homozygous his homozygous wife has attached earlobes. What happens, or what could happen with their children in terms of the shape of their earlobe? Let's analyze this situation and see what we can come up with. So, the man is homozygous dominant for free. So that automatically is a clue that free earlobes is dominant. So if free earlobes is dominant, we're going to use the letter F as our letter, and he is homozygous dominant, so he's big F, big F. His wife is homozygous for attached earlobes. If she has attached earlobes, she must be recessive, and she must be little f, little f. We automatically know that from the setup of the problem. So here is the man, big F for free earlobes. His wife is homozygous for attached earlobes, so she must be little f, little f. The man is going to produce sperm with only one allele. One type of allele, just like in the pea plant. And those are big F and big F sperm. Okay? So he can only produce one type. Likewise, his wife can only produce one type of egg relative to this trait, and that is little f, little f. Whoops. So it's pretty simple and very similar to the one we just did. Let's do the let's do the Punnett Square over here. So the Punnett Square for their children would look like this. Same situation, same analysis. Big F, sperm there. Little f, egg here and here. And analyzing it, we get big F, little f. Big F, little f. You always put the dominant allele first, or the capitalization one first. Big F, little f, and big F, little f. Guess what? 100% heterozygous. Heterozygous, 100% chance of having children with free earlobes. They cannot have a child with attached earlobes. They have a 0% chance of a child with attached earlobes. So if any of you have a mom or a dad with attached earlobes and the other parent has free earlobes and they happen to be homozygous, and your brothers and sisters are all free earlobes, you would have a pretty good chance of knowing what your other parent, the one with the free earlobes, what their genotype would be. Okay, there's two possibilities. They could be big F or big F, little f. Okay? Any of you in that situation who have free earlobes and one of your parents has attached when you did your survey? Okay, so those three with their hands up, your other parent, the other parent with the free earlobes, the only way you'd be able to tell whether they were big F or big F, little f is if your brothers and sisters had free or attached earlobes. If they had free lobes, there's a good chance that the other parent is heterozygous. If they have attached earlobes, the other parent is heterozygous. If they have free earlobes, then the other parent is probably homozygous dominant. Yeah, hold on. Let me stop this recording before I answer questions. Okay? Let's stop this. Save it. Okay.